Role of the primitive streak and the node in patterning the embryo. The primitive streak and the node are crucial structures that play a central role in establishing the body plan of the embryo during early development. These structures are particularly important during gastrulation, where they orchestrate the formation of the three germ layers, ectoderm, mesoderm, and endoderm, and define the anterior, posterior, dorsal-ventral, and left-right axis of the embryo. 1. Overview of gastrulation and axis formation. Gastrulation is a process during which a single-layered blastula transforms into a trilaminar embryo with three distinct germ layers. This process sets the stage for subsequent organogenesis and the formation of tissues. The primitive streak and node are key structures that help guide the cellular movements necessary for the establishment of the embryonic axis and the differentiation of tissues. Second, formation of the primitive streak. The primitive streak appears during early gastrulation as a midline structure on the surface of the epiblast, the outermost layer of cells in the embryo. The streak is critical for the formation of the mesoderm and endoderm through cell migration. Key features of the primitive streak. Formation. The primitive streak begins to form at the posterior end of the embryo and extends toward the anterior. It serves as the structural foundation for the primitive node. Role in axis formation. The primitive streak defines the anterior-posterior axis of the embryo and is central to the establishment of the dorsal-ventral axis, back to belly orientation. Mesoderm and endoderm formation. During gastrulation, cells move inward through the primitive streak and form the mesoderm and endoderm layers. This migration is a key event in the process of invagination, where cells move from the outer layer, epiblast, into the deeper layers. Third, role of the node in patterning. The node, also known as the Henson's node, is a cluster of cells located at the anterior end of the primitive streak. It serves as a signaling center that regulates important developmental processes. Key features of the node: notochord formation. The node plays a critical role in the formation of the notochord, a rod-like structure derived from mesodermal cells that helps define the dorsal axis and serves as a precursor to the vertebral column in vertebrates. Neural induction. The node signals to the overlying ectoderm, initiating the process of neural induction, which leads to the formation of the neural plate and eventually the neural tube. This is the first step in the development of the central nervous system. Axis specification. The node contributes to the patterning of the left-right axis, ensuring that the heart, liver, and other organs are positioned correctly along the body. 4. Molecular mechanisms regulated by the primitive streak and node. The primitive streak and the node regulate the establishment of embryonic axis through a variety of signaling molecules and transcription factors. Key signaling pathways involved in this process include A. Nodal signaling. Nodal is a member of the TGF-beta family of proteins and is crucial for the formation of the mesoderm and endoderm. It is expressed at high levels in the node and primitive streak. Nodal signaling activates the transcription of genes that are responsible for mesodermal and endodermal specification and regulates the left-right axis patterning. Nodal gradients in the primitive streak guide the movement and differentiation of cells into the mesoderm, muscles, bones, circulatory system, and the endoderm, digestive tract, lungs. B. Sonic hedgehog signaling is a key signaling molecule secreted by the node and is involved in the development of the notochord and neural tube. It also plays a role in establishing the dorsal-ventral axis of the body and helps pattern the left-right symmetry of the embryo. Signaling helps establish the floor plate of the neural tube, a critical structure for the proper formation of the central nervous system. C. WNT signaling. WNT signaling is involved in regulating anterior-posterior axis formation and mesodermal differentiation. WNT gradients are established in the primitive streak, guiding cells to form different regions of the mesoderm and ectoderm. This signaling pathway also helps to regulate cell polarity, cell migration, and epithelial to mesenchymal transition (EMT), which are essential for gastrulation. D. BMP bone morphogenetic protein signaling. BMPs play a significant role in establishing the ectodermal fate, the outer layer that forms the skin and nervous system, and inhibiting mesodermal and endodermal differentiation in regions outside of the primitive streak. The BMP gradient helps to establish the neural plate and promotes ectodermal differentiation while inhibiting mesodermal differentiation at the dorsal side of the embryo. EFGF, fibroblast growth factor signaling. FGF signaling regulates mesodermal and ectodermal development. It is involved in the elongation of the primitive streak and regulates neural differentiation in the ectoderm. FGF also promotes cell migration and helps pattern the anterior-posterior axis. 5. The role of the primitive streak and node in left-right patterning. The left-right axis is essential for the proper placement of organs in the body. The node and primitive streak help establish this axis through the following processes. A. Left-right symmetry. The node secretes lefty and nodal proteins, which establish a gradient on the left side of the embryo, ensuring the proper asymmetry needed for the formation of organs such as the heart and liver. Cilia present on the cells in the node help create a fluid flow that establishes this asymmetry. B. Organ positioning. The node's signaling activities guide the positioning of major organs along the left-right axis. For example, the heart normally develops on the left side and the liver on the right, with this pattern being established during the early stages of development. Sixth conclusion. The primitive streak and node play indispensable roles in establishing the embryonic axis, forming the germ layers, and initiating the processes of cell differentiation and morphogenesis. These structures set the foundation for the development of the body's complex organization, including the nervous system, musculoskeletal system, and internal organs, by regulating signaling pathways such as nodal, SH, and WNT. The primitive streak and node are central to the proper patterning of the embryo and the formation of functional tissues and organs. Understanding their roles is critical for comprehending normal embryonic development and the causes of developmental diseases.